and welcome back to day 2 of Invest in Your Planet Career Speaker Series, presented by Palm Beach International, Boat Show Gives Back, and proudly supported by Waste Management and Keep Palm Beach County Beautiful. My name is Lauren Henderson, and I am the Director of Secondary Programs for Junior Achievement of the Palm Beaches and Treasure Coast. Now, I'm very excited to be here with you today as I travel across Palm Beach County and the Treasure Coast to learn more about the job opportunities in the marine industry. We're going to hear from experts all week in the marine industry to share their jobs, their career stories, so that we can all learn more about what's here in our backyard. Today, we are here at WildPine Laboratory, Loxahatchee River District in Jupiter, Florida, and we have Miss Susan Noel, the laboratory manager, and her partner in crime, Mr. David Porter, to tell us more about what they do and how they're keeping the community informed on what's going on within our waters. So go ahead, guys, take it away.
This is our lab. Like you said, we're located at the Regional Wastewater Treatment Plant. Thank you for joining us as we talk about water quality on this great Earth Week. My name is Susan Noel. I'm the lab manager. This is David Porter. He's a lab technician with us, and we have been working for Loxahatchee River District for 25 years. Dave, can you believe it?
No.
Yeah. When we started here, Dave was working part-time, so we had 3.5 people working here. Dave was a half. He was schlepping back and forth between the Marine Science Center and the lab, and I came on board full-time 25 years ago as a lab technician. So since then, I learned, all the tests that needed to be done for the wastewater plant, and I also got involved in some river research. And I wanted to learn everything, so I took on the quality control program. Then with that, I ended up getting a promotion to assistant lab manager, and then with the expectation that when the lab manager retired, I would be ready to fill her shoes. So I've been lab manager for 10 years. So what my background is I have a BS degree in biology. It was quite a long time ago, so there weren't as many options as the students have nowadays. Some of our interns that work with us have marine biology degrees, environmental science degrees, geoscience degrees. There's a lot of different specialties. I don't know how important it is to specialize in a certain degree. I guess it depends on if you really know what you want to do or not. But biology worked out for me, and it's still available. I think the more important thing is the courses that you're taking. Obviously, getting a degree is important for a marine some kind of marine career, but also the courses that you take are biology, chemistry. I took a lot of chemistry courses. We do a lot of chemistries in this lab. Also, statistics is a very important part of our job. And GIS is also a very important part of our job. As you'll see, we use when we produce maps, we use GIS. When we write reports, we use a lot of statistics, and we make charts. Also, we have some special skills that are required in this kind of field. So if you're really interested in working in the water, you would want to practice your swimming skills, your snorkeling skills, your boating skills, and possibly considering getting a dive certification. So we have a very important mission to protect and preserve the Loxahatchee River. And also public health is a very important part of what we do, as you'll see later. The ways that we support our mission is we do wastewater innovations. For example, we've converted all our septic tanks into sewers. That's very important thing to do. We do environmental education. We do a little bit of environmental education here, like for instance, today, but we also have a river center that does the majority of our environmental education. Our main focus, besides the wastewater plant, is to do scientific research. Do you know that about 40 years ago, wastewater was seeping into the Loxahatchee River? So we had, at the time, we had some very good community leaders who had the foresight to build a wastewater plant and then convert all the houses in the Jupiter to Cuesta area from septic tanks to sewers and reroute that water to the wastewater plant. So with that, the lab was developed because we have to do a lot of wastewater testing for the Florida Department of Environmental Protection. And so if you want to walk this way, one of our lab technicians, Danny Tomasello, does a lot of our daily wastewater analysis. And so here you can see he's stirring. What are you doing, Danny?
So I was doing suspended solids on our wastewater. We read it before and after. It goes through the plant and the whole process. To make sure that it's within DEP's permitted amount of suspended solids that are allowed.
Right.
And then we also do biological oxygen demand tests. That also allows us to know when we send the water out to the public, it goes to our golf courses and everywhere else for irrigation, if it will deplete the oxygen in the lakes that we send it to. We need to know that. We don't want to kill any fish or anything like that.
So how long have you been working here, Danny?
5 years.
Danny is our youngest employee.
Yep.
We actually have 6 people working in the lab. We have 4 lab technicians, Danny being one, Dave being another, and there are 2 others floating around. We have a PhD senior scientist working with us who's not here right now, and myself. And then, of course, we have a part-time intern as well. So with that, like I said, the lab is a very important part of our scientific research. So I would like to share with you some of the current projects that we are working on. So the first project is a long-term water quality monitoring project. We call it our Riverkeeper project. So we have about 50 sites in the river, and we go around on a regular basis and we take water samples. Now, some of the sites we can get to from our boat, and Dave is our boat captain. So he takes us up the river. We start at the Jupiter Inlet, we go all the way up to Trappers. I don't know if you know where that is, but it's up in Jonathan Dickinson State Park. We have another group of people who drive around in a truck and take water samples in the tributaries and canals where you can't get to by boat. So what we do when we get to a site is we use this multi-parameter water quality instrument, and we put it in the water, and it can detect different parameters. These field parameters are very important. They're the kinds of tests that you have to do in the field, because if you fill up a bottle and bring it back to the lab, the result's going to change. So what are some of these tests? We have a probe for pH. We have a probe for salinity. We have a probe for dissolved oxygen. And we have a thermometer in there so that we can record the temperature. So we can look at the instantaneous readings on this meter, and we can record the data on this data sheet. And we can store the readings in the meter and record them on the data sheet. You're probably thinking, why do we want to do both? Well, we're in a world of redundancy here. We know that things can happen to electronic equipment when you're out on a boat working in salt water. So we write all the readings down. Also from experience, we know that you can lose your clipboard when you're in the field or it could get rained on or it could fly off the boat. So we have, we have plan A's and plan B's.
Anything can happen.
Yeah. So the field readings they're important, but they're not the meat and potatoes. What else we do is we collect water samples and we bring them back to the lab and we analyze them for, I call them the big four. We have bacteria. We have 3 different bacteria tests that we analyze for. We analyze for total nitrogen, total phosphorus, and chlorophylls. Dave does the total phosphorus and the chlorophylls analysis. So what do we do with this data? Well, we make it available on our website. We use GIS to publish spatial maps. We use statistics to create charts and graphs, and also to write reports and hopefully some peer-reviewed journal articles. So we'll be showing you links to all this data later on in the video. The other
Nutrients?
That's all for Riverkeeper.
Yep.
Now I want to talk a little bit about our seagrass program. So we have a long-term seagrass monitoring program that we do on a regular basis. And we have a grid that we use to collect data with. So this grid is 1 square meter. It's this is the standard operating procedure that most agencies use for seagrass. So we can share data and talk about what's going on in the world. So this is 1 square meter. It's divided up into 100 smaller cells. So we can very easily come up with a percentage of how much seagrass is there. So what you do is, first of all, this is a project where you need to get wet and you need to be comfortable working under the water. Whereas on a boat, you're typically dry.
Right.
Yeah. So what you do is you put the grid down over your seagrass bed. We have 5 different seagrass beds that we routinely study. And each seagrass bed is divided up into about 30 random points that we throw a grid down and collect data for. So the types of things that we're collecting are on this waterproof paper. So we're going to count all the squares that have seagrass in them, and we're going to count all the squares that are bare or have sand in them, and we're going to record that. And then that gives us like a percent occurrence of how much seagrass is there. And we do that for total seagrass, and we do it for each seagrass species. We have 5 different seagrass species right now that we are documenting. 2 of them are paddle grasses, 1 is shoal grass, 1 is manatee grass, and 1 is turtle grass. So after we get a percent occurrence, then we try to estimate a bacterial density, which is a little more subjective and takes a little bit of training to do. And then after that, we also count shoots of seagrass. So yes, when my family says, "What did you do today?" I said, "Well, I was counting grass." And they're like, "And you got paid for that?" So we count the grass, and then we also measure the grass. And we have these waterproof field sheets that we record all our data on. And this data goes in our database. And we do the typical things that we did with the Riverkeeper project, where we make maps and charts and write reports.
And we can monitor the increase in densities. We can monitor the change in the seagrass growth and spread over time.
It's another long-term database. Yeah. Yes.
Yeah.
All right, now we would like to talk about our third program that we're involved in. It's called our weekly water quality bacteria program. So you probably have heard of the Florida Healthy Beaches program that Palm Beach County and other counties are involved in, where they test different beaches, and they issue advisories based on the amount of bacteria that's in there. Well, we have our own program, and it's just for the Loxahatchee River. We have 10 stations that an intern drives around and collects samples at. And this project was actually started by an ex-intern. The way that the project started was she wanted to go play on the sandbar near her house one day. And she's like, geez, I wish I knew what the water quality was. And I'm like, well, we can test it and tell you what it is. So we did, and then we're like, wow, this is really cool. We should like expand this program. So we did, with her help, we picked out these 10 stations in the Jupiter area that people frequently recreate. DuBois Park is one of them. And we'll show you a map in a little while of where all these are and how you can get the information. And so we test for enterococci bacteria. Now that's the indicator bacteria that the CDC and the EPA have decided that is the best indicator for safe water. Why do we want to do this? Well, when these bacteria are present, and you swallow the water, they can cause gastrointestinal illnesses like diarrhea, vomiting, nausea. You can get a fever. So, it's really important, especially for parents when their kids are swimming in the water, because young children will be most affected by this, and also the elderly and the immunocompromised people. So it's important information for them. A regular healthy person may be okay, depending on what the quantity is. Also, it's an indicator organism, so if that's present in high concentrations, then chances are there are going to be other dangerous things present as well. But this is an easy, inexpensive test for labs to do.
Now, this test is only in the Loxahatchee River estuary, in the North Palm Beach County area. So wherever you guys are located, those results that we get may not be indicative of what the conditions are where you're at. Now, that's probably important to mention.
Right. There's 3 main ways that bacteria can enter the river. One is through human sewage. Well, we pretty much have that covered. We have a wastewater plant. We converted all our septic tanks to sewer. We have a very high functioning wastewater plant. So we can pretty much rule out the human sewage unless, there's like, a small, a baby diaper or something like that, or, a homeless population or, something like that. But we can pretty much rule that out. The other way bacteria can enter the river is through our pets and wildlife. So we can control our pets' waste by picking it up and disposing of it properly. We can't really do a whole lot about wildlife. I'm talking about birds, raccoons, that type of thing. That's contributing to the fecal matter. The third way is through stormwater runoff. So that can be controlled somewhat. Stormwater, we need to educate the public to not dump their grass clippings and leaves on or in storm drains. And their garbage. I mean, it's not a garbage can. So when the organic matter decays and we get a big rain event, guess what? It's all gonna get washed into our rivers and oceans. So we can control that somewhat. But those are the 3 ways that, fecal contamination can happen in the river. And that's why it's important for us to monitor and get the word out to the public as to what the condition is that week. We feel like a weekly basis is, now the water's constantly moving and changing around, but we can at least let the public know on a weekly basis what's going on in the waterway. Now we would like to...
let's show them how we do it.
Yeah.
Okay.
Okay, here we go. We're gonna head into the microbiology room. So this is where the action happens. So first we have to sample. So like I said, our intern will drive around and he will either collect a sample by dipping the bottle into the water or he will use a sample pull if that's more practical. We're not going to show you that part. We have our sample already collected. Then we bring it back to the lab, and we're always recording times and dates, and we're recording the temperature.
Right.
The sample is collected on ice, so it takes us about a couple of hours to drive around town, so we always have to put the samples on ice after they're collected. So when we get back to the lab, we need to check the temperature of the samples to make sure that they are starting the cooling process.
What the ice does, just very, very briefly, is it stops the reproduction of the microorganisms. So you get a more representative sample of what you've collected in the waterway. So you collect in the waterway and you put them immediately on ice to stop any more growth or increase in the population of the bacteria inside your sample.
Right. And now show them what you do when, how do you set up the test?
Well, first thing we're going to do is bring the samples into the lab. We're going to monitor times.
We're ruled by the clock in the lab.
Yes. So
everything has a time associated to it.
Yes. So we jot down the time that it comes into the lab, what time we start setting up the test. We also are very cognizant of what the temperature is of our sample. So we'll use a little digital thermometer, quickly grab a temperature, jot that down, make sure it's within limits. So then the next thing we're going to do is actually make a dilution of that sample. So in general practice, we will take a saltwater sample and dilute it 1 to 10. So we'll take 10 mils of our sample.
And the reason why we're doing a dilution is because it's the standard operating procedure from the manufacturer that says if you are testing a saltwater sample, there's going to be interferences from the sodium, calcium, magnesium, potassium, cations. And so we have to do a dilution on our sample to take out the interferences. And then he's going to dilute that back up to 100 milliliters. The reason why we collect 100 milliliters and test 100 milliliters is because the CDC said that's about the amount that someone's going to drink. It's a small glass of water.
Right.
So he's doing his dilution, and then he's going to add the chemical, which is it's called Enerolert, and it's manufactured by IDEX, and it's a nutrient indicator. So we're going to shake it up until it dissolves. And so after we incubate the samples, if there's enterococci in there, they will produce an enzyme, and the chemical will cause the bacteria to fluoresce under UV light. So when we show you the results, you'll see blue fluorescence. So it's a very easy test to do.
So it does take a little time for this to dissolve. So we have one already prepared. Now, I know this should be done under sterile conditions. So there's no cross-contamination. I know I lost my pipette tip, but this is just for demonstration purposes. So this is the one that the Enerolert has already dissolved in. And then we add this to our tray.
Those cells in that tray are like individual test tubes. So we're going to count the positive cells after incubation.
So
we didn't
warm up
the sealer, so it's not going to seal right now.
So we will run this through the sealer. It will seal our tray, and the tray will look like this when it's all nice and sealed. Individual tubes. So once it's sealed.
So let's move on to day 2
Yep.
because we're running out of time.
I know. So we'll put these in the incubator. It's a 41.5 degree incubator for 24 hours. Once that's done, we'll come back in take our sample out,
You're going to take both of them out.
take them both?
Mhm.
All right. And then we use a UV lamp to look at the results. Let's see. Can you guys zoom in and see that? So if a well has fluoresced to blue, that's a positive result. There we go.
So it looks like we have 1, 2, 3, 4, 5, 6, 7, 8, 9 on that one.
Mm-hmm. Yep.
So what does that
translate
equilibrate to in concentration? So we have a chart that has been developed. It's part of the standard operating procedure. So for the number of wells positive, we have a 9. Can you
Yep, sorry.
see it? And that translates to a most probable number of 9.9. Now, we did a 1 to 10 dilution. Here's where the math comes in. So we must multiply 9.9 by 10 to get our final result. So our final result is 99. All right, so what does that mean? So if we compare our result of 99 to the water quality standard of 71, that says that that body of water at the time that we sampled is of poor quality. So it would be colored red instead of green. So that's how you use the data and understand, what the results mean. And we have all this data published. We publish a weekly map. And as you could see, if you go to page 1, last week, all our samples were in the green or good. But this one is just a practice, so we're not going to put it on our website. We're just practicing what a poor sample would look like. So, but it would, if we had published it on our website, if it was a real sample, it would show up as a red dot. And now if you go to page 2, so after you're done looking at the, data for that week, if someone who hasn't been on our website and wants to familiarize themselves with a certain spot, maybe that they recreate, like possibly Dubois Park, they might want to see, hey, how has Dubois Park been doing for the past year? Well, you can look at our table and you can see Dubois Park up here with all the weeks that we tested. And in the past year, you can see we had maybe 3 issues where there were swimming advisories. But you can see that there are some other sites that we picked out that are actually worse than Dubois Park.
Okay.
So we just wanted to thank you so much for attending. We're out of time. We're going to put some contact information up with my contact information for questions, website links. And I also just wanted to really quickly say that we do have internship and volunteer programs. All the information and applications are available on our website. There's a few qualifications. You must be 18 years of age. You have to be currently taking college classes or a recent college graduate. And we hope to see you in the future.
Yeah. Yes, thank you guys. Now, I was trying to be as detailed as you guys are with your note taking. I was looking at all the charts that you guys take, and I was like, wow, you
There's
really
so much.
need to be detail oriented. You obviously need to be able to work well with others.
Absolutely. You have to be an individual and a team player for sure, because when we do these projects, you can't get things done by yourself. Like our PhD senior scientist, he develops these research projects, but he can't do it on his own. You need a team of people who know what they're doing. Yeah.
Well, listen, you guys are doing such important work, and the way you inform the community members about, you inform the public about what's going on in our local waters. And I will definitely be looking at this website this summer as I get ready to take my kids out to the different locations for recreational activity in the water. So thank you guys so much.
That's
you guys are absolutely
That's so great to hear.
today.
Thanks for coming by.
Yeah.
Thank you guys so much.